I think we are going to start today. I would like to welcome everyone to this webinar. And before we start with the webinar, I just want to introduce you to our society. Maybe you don't know it. So we are, or I'm Cajochen from the University of Basel, also a board member of the Society for Light Treatment and Biological Rhythms. And I would all of you encourage to become a member, because it's a really interesting society bridging basic science with clinical science in terms of circadian and light research. So we are going to have a virtual meeting very soon, our annual meeting, May 19th. So you see it on this slide. And just to let you know, the registration is going to be sent out tomorrow, hopefully, and it's for free. So you don't have to pay anything. It's for free for non-members. And also for members, of course. But the talks, you can all, afterwards, you can see the talks only, this is only allowed for members. And just to let you know, now it's really the time to join our society, because you become 2 years for 1 year. And you see here, the membership fees are really not too high in my view. So you can go to our website, SLTBR, and see all the details, where we are, who we are, and how to become a member. So this was the advertisement at the beginning of our webinar. And now, of course, it's my really great pleasure to introduce today's speaker, Dr. Diego Fernandez. Diego did his PhD on the pathophysiology of the visual system at the University of Buenos Aires in Argentina. And after his PhD, he moved to the US, where he became a postdoctoral fellow at Johns Hopkins in the group of Samer Hattar. And as you can see from this slide here, he's at present a staff scientist in the section on light and circadian rhythms at the NIMH. Well, Diego has a fascinating CV in my view. He's interested in understanding how environmental stimuli actually affect subconscious processes that eventually guide our behavior. And as you may notice, light is probably a very strong and very important environmental stimuli, and light, or the non-visual effects of light, I think became one of the major research topics in his excellent studies. Well, I personally became particularly interested by his work, or impressed by his work, where he discovered a novel retina-brain pathway that involves the perihabenular nucleus of the thalamus. And as you know, this nucleus regulates mood behavior in mice, but it could also be an interesting pathway when you are looking at the clinical effects of light in humans. So Dr. Fernandez received numerous awards and honors. Just to mention two: the Excellence and the Merit Award on Research on Biological Rhythms by the SRBR. And I'm sure you came across his work in the literature. He has published very innovative work in high-impact journals such as Nature, eLife, and also PNAS. So for all these reasons, I'm very happy, actually, Diego, that you agreed to give us this webinar today. And I think we are all looking forward to your talk. Thanks again for joining in, all of you. Just to let you know, if you have questions, put them in the chat, and Renske is going to collect them, and we are going to discuss it at the end of Diego's talk. Now let me unshare my screen, and then you can start.
Perfect.
Okay.
Okay. Great. Thank you very much for this nice introduction. Thank you for the invitation. It's a real honor and a pleasure to be here today, and thank you everyone for connecting to this seminar. I really enjoyed preparing this talk, and I hope this would be useful for you as well. I would like to discuss today recent research that studies the mechanisms and the circuits underlying the distinct and diverse effects of light on mood, learning, and metabolism. Throughout my career, I've been always interested in understanding how the changes in the environment are represented in the brain and how this representation affects conscious and subconscious behaviors. In any sensory modality, the perception starts with a receptor that detects the stimulus. Then it activates different cellular transaction pathways, and through neural networks, it finally leads to the perception. But importantly, this information is always compared with previous experiences that are all embedded in a cognitive map. In a way that, in this example, we have a pure visual perception where we can see colors and shapes, but this image can also evoke memories and emotions that are associated with it. In mammals, the retina is the only organ that encodes light information to affect brain functions. And it follows the same principles that I described before. Photoreceptors at the back of the eye transform photic information into electrical signals, and then through interneurons, the information reaches the projection neurons of the retina, the retinal ganglion cells that route this photic input, this photic information to many brain areas. As mentioned, light is critical for visual conscious functions, and there are many areas involved in the image forming functions, as well as movement detection and integration between light and other stimuli. As an example, the superior colliculus integrate sounds and vision and help us to orientate in space. We also know that, on the other hand, changes in light intensity are also captured by the retina, routed to the brain, generating an internal representation of time that synchronizes circadian rhythms with the external LiDAR cycle. And this process is known as photoentrainment. In particular, these rhythmic processes are all orchestrated by a central pacemaker housed in the suprachiasmatic nucleus of the hypothalamus. And as a central clock, the SCN controls and coordinates the function of rhythmic functions and peripheral clocks located throughout the body. Remarkably, the cells from the retina that drive photic input to the SCN constitute a subpopulation of atypical retinal ganglion cells. They are similar to conventional ganglion cells involved in visual functions, but they also express a photopigment called melanopsin, making these cells intrinsically photosensitive, or iPRGCs. And this population of cells were identified about 20 years ago and were originally considered an homogeneous population of neurons that blend photic input from photoreceptors as well as the intrinsic mechanisms and project mostly to the SCN. Further studies have revealed that, in fact, it's more complex than this, as expected, and there are at least 6 different subtypes of iPRGCs. Studies evaluating anatomical and electrophysiological properties of these neurons revealed this diversity. And as you can see in this model, there are also different levels of melanopsin. So we have now a good understanding of the connectivity of this individual subtype with different retinal cells. However, another interesting aspect that we have just started to understand is the diversity and complexity of iPRGC projections into the brain. To study this, we developed a conditional tracing system that allowed us to identify iPRGCs at the single cell resolution. As shown in this image, we were able to induce the expression of the reporter gene, the alkaline phosphatase, in a single iPRGC. And we can then identify the dendritic arborization, distinguish the cell type, but also follow the axonal projection all the way to the optic disc, to the brain, and the targets. As expected, we found innervation to the SCN, but we were surprised to see the complexity of a single cell innervating the central pacemaker. This is a 3D reconstruction where you can see a dense and bilateral innervation to the central pacemaker. And when we compare different cells, we got a beautiful map of connectivity from retina to the central pacemaker, such as in that individual cells has particular connections with distinct subdomains of the SCN, also such as in that different aspects of circadian functions could be modulated by different connections between the retina and the central pacemaker. But furthermore, we found that in all cases, cells projecting to the SCN, they have also collateral projections that innervate multiple brain areas. In the example that I'm showing you here, a single iPRGC, in this case it's an M1 cell, send dense projections to the SCN, but also it has collateral innervation to areas of the hypothalamus, as well as regions of the geniculate complex and the superior colliculus. Together with a massive amount of data, different labs have contributed to this. We have now a better and a current understanding of the description of the complexity of these circuits where the retina can affect multiple regions. And as you can see in this model, it goes beyond the SCN. And interestingly, these iPRGCs project to areas that are known to control subconscious functions, suggesting that changes in light driven by iPRGCs can directly modulate the function of these regions. And this becomes particularly relevant in the context of abnormal light exposure. Today, a large fraction of world population is exposed to artificial light. And it has been shown that the chronic exposure to artificial and particularly abnormal lighting conditions, such as those experienced under shift work schedules, are associated with multiple signs of physiological and behavioral deficits. So based on the widespread brain innervation of iPRGCs that I described before, the conceptual framework of the research that I will discuss in this talk is that multiple retinal brain pathways drive the diverse effect of light exposure or different conditions of light exposure on physiology. For the first part of the talk, I will focus on recent research focusing on the mechanisms and circuit linking light with mood and cognitive functions. And for the second part, I will discuss whether light routed through iPRGCs can have widespread effects in behavior, affecting, for example, metabolism. So first, what do we know about iPRGCs and particularly about their direct effect on behavior? And I would like to start addressing this question by discussing this elegant work from the Lucas lab, where they explore the direct effect of iPRGCs by using a chemogenetic strategy. In this case, this is a DREAD technology. It's a new technology that basically using adenoviruses, they were able to induce the expression of a DREAD receptor, specifically in iPRGCs. Then with the addition of the ligand CNO, it causes the depolarization, basically the activation of these iPRGCs. What they did was kept animal in constant darkness. So this is a very simple and smart strategy to avoid any light stimulation. But then they added CNO with a single IP injection into the animals, causing the acute and selective activation of iPRGCs. And in these conditions, what they found is a massive activation of limbic areas, different regions, but also they observed widespread changes in behavior, including arousal and anxiety. So this data revealed that acute changes in iPRGCs can drastically affect multiple brain regions. In a recent study from the RAN group, they showed that acute light pulses are also able to modulate complex behaviors. So the strategy is in some way similar to the DREAD experiments that I showed you before, where they cause this acute activation of iPRGCs. But in this case, they use light pulses. These are 2 hours light pulse, 3,000 lux applied to animals housed in normal LD cycles during 3 weeks. And what they found is that under these conditions, this light treatment clearly improves the performance in spatial memory tests. Then they evaluated whether this light treatment could be beneficial in a context of mood alteration. So to explore this possibility, they used a classic model of chronic social defeat, where animals are exposed for about a month to this aversive stimulation where they have both physical and sensory stress. And under these conditions, it's known that animals showed severe mood deficits. What they did next is to expose animals to the same conditions, but the last 2 weeks of this treatment, they applied the light stimulation. similar to the protocol that I described before. This is just 2 hours of bright light per day, particularly early in the morning. And under these conditions, animals showed clear improvement of mood, revealing that in some way light treatment blocks the negative aspect of this stressful condition. So combined, this result revealed that brief activation of the retinal brain circuits, possibly IPRGC's circuits, at specific time of the day, at specific circadian times, not only modulate behavior, but it can also be used as a therapeutic intervention. The question then is what's happened, or what is the impact of applying light pulses at the wrong time of the day? And in particular, what are the consequences of doing this in a chronic way? So this question was addressed in a pioneering study from Tara Ligate when she was a PhD student at the Hatar Lab, where they used the T7 cycle. This is an ultradian LiDAR cycle that presents periods of light and dark during 14 days. What is remarkable about this cycle is that independently of nocturnal or diurnal animals, which is a key question in circadian biology, under these conditions, animals received both light and dark stimulation at different phases of circadian activity. So what they found is that exposing control animals under these regular or abnormal light conditions, they found that the irregular protocol caused severe learning and mood deficits, whereas these alterations are completely independent from any sign of circadian disruption. And it was possibly the first demonstration that circadian alterations can be dissociated from mood and learning deficits. Next, they studied what are the role of IPRGCs in mediating these effects. And to test this, they used animals where IPRGCs were genetically ablated. Most of the cells are not longer present in adult animals. They housed animals in these two conditions, and what they found is that the chronic and irregular light exposure do not affect behavior. Animals have normal learning, normal mood, and free running responses, revealing that IPRGCs, this is possibly the first demonstration that they are sufficient for driving all these effects, and particularly drive the deleterious effect of irregular light exposure. Further, we explored the mechanisms that could be mediating these responses. And we recently revealed that the direct effect of light on both mood and cognitive functions are driven by distinct and parallel retinal brain circuits. So we first evaluated to what extent the IPRGC SCN circuit affects behavior. So to address this question, we took advantage of a mouse line generated in the Hatar Lab, where most IPRGCs that express the transcription factor were ablated by the expression of the diphtheria toxin, except for a small population of IPRGCs that are brain3B negative that exclusively innervate the SCN. So by using these animals, we have now a perfect system where we can study the direct effect of light through IPRGC affecting the circadian pacemaker and the SCN. These animals were housed under regular and abnormal light conditions for 2 weeks, and then we measured the behavioral consequences. As shown here, we first observed that animals housed under abnormal lighting conditions showed learning deficits, as shown here with the lack of preference for the novel object. This is a powerful tool to evaluate short-term memory. We found also alterations in long-term memory using a variety of behavioral tests. But what was remarkable was that under the same conditions, these animals where IPRGC only innervate the SCN do not show any sign of mood alteration. So the first conclusion here is that the IPRGC SCN circuit is sufficient for driving the negative aspect of light on learning and memory. However, this circuit is not sufficient for altered mood, indicating that there is another circuit that could be mediating these effects. And among all candidates, we identified a region that is well positioned to link environmental light to affective behavior. This area is the perihavenular nucleus. The PHB It's a nucleus that is part of the dorsal thalamus that receives dense innervation from retinal cells, exclusively from iPRGCs. We also show that there are acute changes in neural activity when light is also changing. And finally, and importantly, the PHB is interconnected with mood regulating centers. Using a variety of tracing strategies, we found that PHB neurons have collateral projections to mood regulating centers, including the ventromedial area of the prefrontal cortex, dorsal striatum, as well as the nucleus accumbens. So next, we decided to explore and confirm the role of the PHB in mediating mood changes associated with changes in light environment, in environmental light, sorry. So to do this, we set up different strategies to selectively manipulate this region. We did different tests, and I'd like to describe today one of them, where we use a chronic strategy to silence the PHB neurons. Using AV injections specifically into the PHB, we were able to induce the expression of the diphtheria toxin exclusively in PHB neurons, and it would cause the silencing of these neurons between them and connections with their targets. These animals, a few weeks later, were exposed to the irregular light cycle that is known to affect mood, and what we found is that the chronic silencing of PHB neurons was sufficient to block any alteration in mood. As I mentioned before, we perform a variety of behavioral and manipulation strategies to confirm that the PHB is both necessary and sufficient for driving the deleterious effect of light on mood. So together, this body of literature suggests that the circadian timing of light exposure has distinct effects on behavior, and furthermore, it's possible that this effect may be mediated by different circuits. Today, I just described time of day of light exposure, but another features of light are also critical to address these important questions, and hopefully this could be part of the future research. Some questions are, what is the importance of the intensity of light as well as the spectrum of light exposure? As mentioned, we described different circuits mediating the chronic and negative effect of light. Recent data have also characterized the circuits involved in the acute effect of light treatments. And a future question, future line of research could hopefully study whether these circuits integrate photonic information to drive a coherent output. And importantly, since we are talking about different circuits, it could be essential to discriminate whether these circuits involve discrete functions, discrete alterations in behavior, or whether they have widespread alterations. Some of these questions were nicely reviewed in these two articles. I encourage everyone to read them. They're really cool reviews. An aspect that I'm particularly interested in is whether additional symptoms, as mentioned, are associated with chronic exposure to irregular light conditions, could be also mediated by independent retinal brain circuits. And among them, a higher incidence of metabolic alterations have been extensively documented among populations of shift workers. What is important to mention here is that these working schedules not only disrupt the regular light cycles, but also causes misalignment between biological and social cues. At this point, it's important to describe these photic and non-photic time cues affecting circadian functions. Under these conditions, we have, for example, a complete misalignment between light cycles, feeding cycles, as well as sleep-wake cycles. In addition to light, we know that changes in food availability are known to affect circadian functions. And as shown in this diagram, it was proposed that these changes in environmental conditions could impact multiple aspects of physiology by affecting first the central pacemaker. We know that light can directly affect the SCN through iPRGC projections, but we have we're just starting to understand the potential pathway mediating changes in food, for example, to affect circadian functions. And we can expand this question to any non-phoric time cue, social interaction or stress or food, as shown here, can all of them have a direct impact on circadian functions. So in a recent study, we explored the possibility that photic and non-photic cues, such as food availability, are integrated within the circadian system to drive a coherent output. The question here was, in addition to these parallel pathways, is there any point within the circadian circuit where these distinct photic and non-photic time cues are being integrated, having a coherent output under normal and, of course, abnormal lighting or environmental conditions? So to test this possibility, we explored first, and this was a proof of concept in some way, we explored whether the lack of IPRGC innervation affects circuits driving circadian responses to non-photic cues. In particular, we used animals where IPRGCs, all of them, were ablated at early time points during development, and we then tested adult animals. In this study, we also use enucleation strategies to mimic some of the alterations caused by lack of IPRGC innervation. And in particular, we use a non-phoric entrainment paradigm that is a classic method in chronobiology defined as time-restricted feeding. Briefly, under these conditions, animals are housed with food ad libitum for a couple of days, weeks, and after that, the access to food is limited to 7 hours per day. In this particular case, animals were housed in constant darkness, and this is extremely important for us. So in this case, food availability constitute the only recurrent time cue for these animals. There is no photic stimulation in these experiments. And what we found, and what was previously found, and this is shown in this nice study, under these conditions, animals are first in free running condition, of course, but then when access to food is limited to these 7 hours, animals start showing this powerful anticipatory activity. You can see how they displayed robust and sustained locomotor activity prior to food access. So we used this paradigm, and we decided to test whether the lack of IPRGC innervation could impact these non-phoric properties. And we found that although there were no changes in total food consumed or body weight, adult mice lacking IPRGC from early developmental stages showed clear deficits in the anticipatory responses to restricted access to food. In this model, you can clearly see how animals show this free running period that is completely unaffected by changes in food. But as I mentioned before, feeding activity was normal, showing a complete dissociation between feeding activity from the circadian component associated with the timing of food access. This data is quantified here, where we found that both animals show no anticipation a few hours before food access, but at 3 hours before, they have this huge increase in anticipatory activity that was significantly affected in animals without IPRGCs. So these results combined suggest that the lack of IPRGC innervation affects circadian centers, driving these non-phoric responses and these non-phoric entrainment properties. Next, we decided to identify and explore the potential circuits driving these responses, and we therefore explore photoresponsive areas that are known to also sense changes in non-phoric cues. In particular, we focused on the circuit that connects the IGL with the SCN. The IGL is the intergeniculate leaflet, that it's part of the geniculate complex, and similar to the SCN, is densely innervated by IPRGCs. It was previously shown that changes in light induces acute activation of cells, both in the SCN and IGL, demonstrating that these two areas are photoresponsive areas. And in addition, it was shown that the IGL connects with the SEN through the projections of MPY expressing neurons. In fact, the MPY innervation to the SEN is exclusively coming from IGL neurons. So we decided next to evaluate whether this circuit could be affected in conditions where we removed all IPRGCs from early development. IGL is known to also respond to changes in food. As shown here, models of food deprivation showed clear changes in the activity of IGL, increasing also the levels of MPY. So this model is a perfect candidate for integrating photic and non-photic cues from the environment, affecting then the circadian pacemaker. What we found is that under normal conditions, animals lacking IPRGCs from early development, and these are all adult mice, showed drastic alterations in the IGL-SEN circuit. As shown in this example, there is a significant reduction in the levels of MPY at IGL levels. However, the number of cells was constant. It's just alterations of the peptides. And moreover, we found similar and drastic reduction in MPY levels at SEN at the level of the terminals of these IGL fibers. As I mentioned, these mice were housed under normal conditions. Therefore, we then tested whether these alterations could impact the functional connectivity and the responses to changes in the environment. To do this, we use again the time-restricted feeding. We expose animals to constant darkness with food ad libitum, and a few days later, food was limited to 7 hours time window per day. Under these conditions, control animals showed these robust anticipatory responses, and 3 weeks later, once we confirmed the anticipatory responses, we measured the C-false induction at the time when animals are expecting the food, so prior to food access. And what we found is that control animals showed this robust activation of the IGL, particularly measured with the C-false induction. Under ad libitum conditions, the number of C-false is almost 0. So there is a drastic increase in neuronal activation that is associated with the expected feeding time. And what we found is that animals lacking IPRGCs, in this case, the IGL is largely unresponsive to changes in food access. Again, in normal conditions, the IGL shows really low number of C-false. So together, these results revealed that the early IPRGC innervation is required for the proper assembly of this IGL-SEN circuit, which showed clear responses to changes in food access, and moreover, drives anticipatory responses to time-restricted feeding. I'm not showing you today all the data from this paper, but we use strategies to specifically manipulate IGL neurons, specifically expressing MPY, to the SEN, and we found that the silencing of these cells when animals are showing robust anticipatory responses is sufficient to alter this behavior. Moreover, we found that these alterations are not only affecting behavioral output, but they also have an impact in metabolic variables such as the levels of ghrelin, which is an hormone that is known to be highly induced in response to food deprivation. Finally, we propose here that the IGL-SEN circuit acts as the center, integrating photic and non-photic time cues, guiding a coordinated circadian output. Furthermore, this could have implications on how abnormal light conditions affect metabolism and energy balance. In addition, we discussed today several studies that have characterized these multiple retinal brain pathways, driving different aspects of light affecting behavior. In particular, we focused on the connections of retina with SEN and PHB circuits that are known to drive the deleterious effect of light on mood and learning functions. And this was particularly important in the context of irregular light exposure. I also described data from the RAN lab these two beautiful neuron papers, where in addition to explore the light treatment, they also characterize potential retinal brain circuits mediating these direct effects. And interestingly, they found that projections from the retina to the geniculate complex are then connecting these cells with either the hippocampus or the lateral habenula. And these circuits are potentially mediating the beneficial effect of light on different models, either alterations of cognitive functions as well as mood deficits. And finally, I would like to mention that when we think about mood disorders in general, when we think about major depression associated with changes in light, it's important to consider that these pathologies are characterized based on a variety of symptoms. However, we only have treatments that are designed for the whole pathology without considering the specific aspects of these symptoms. This data, of course, working with rodents, it's in some ways sufficient to prove that many pathways can be differentially affected by light. And depending on lighting conditions, depending on the time of the day, how chronic this exposure are, it could have differential effect on these circuits, finally having differential effect also on the symptoms, and therefore it could be extremely helpful in designing better treatments. In addition, these are just 3 examples of how light can in parallel affect mood, cognitive functions, as well as metabolic functions. And we can expand this analysis to any other brain circuits, as mentioned before, with the many targets that were recently discovered for IPRGCs, asking the question whether other symptoms, other behaviors, or physiological functions could be directly modulated by changes in light through projections from retinal ganglion cells, or particularly through IPRGCs. With that, I would like to thank, first of all, Samer Hattar, my mentor, and the director of the section of light and circadian rhythms, and all people in our lab. The data that I showed you today from our lab was part of the research that we started at Johns Hopkins in collaboration with Reishi Kuruvilla and Haichin Sao. And now we're working at NIMH with this fantastic group of people. And during all this time, we have also fantastic collaborators. I mentioned here the work that we did in collaboration with Mario Penso, and previously working in collaboration with Alfredo Kirkwood. He helped us with electrophysiology, David Burson lab at Brown University, and Alan Chen, who was leader on the study that I showed you at the beginning with the projections of individual IPRGCs to the brain. Finally, I would like to also thank the Pew Foundation for the support during my PhD. I encourage everyone to apply if you can. And with that, I'd like to thank you all for your attention. And I think that we have time for questions.
Well, Diego, thank you very much for your fascinating talk. It was really a pleasure to listen to you. And I'm now very unpolite. I'm going to ask the first question. But we have questions in the chat, and Renske is probably going to, she's going to moderate it. But just because I'm also interested in sleep, and you mentioned sleep at the end of your final remarks, don't you think that sleep is a major modulator of your effects, especially in your first study? I mean, in the T-cycle study, as you know, sleep can change mood, can also change, cognitive processes. So if you change sleep, you also change your other output variables. What do you think about it? Do you have some data on how you change their sleep pattern there? Exactly, yes.
Yes, that's an excellent question. At the time when Tara and Samer published this paper, they used the T7 cycle, particularly because there are no drastic changes in circadian function. They found that rhythmicity of animals is preserved. The expression of clock genes was also similar to controls. And the release of hormones was also rhythmic. And they also measured sleep architecture. And they found that the amount of sleep is not significantly affected. However,
well, maybe it's not the amount, but it's more the, You know what I mean? It's like the amount of sleep across 24 hours may be the same, but the distribution is certainly different, right?
It's absolutely. This is a possibility, and it could be really, really cool to explore and have a deeper analysis of sleep conditions in these animals, sleep architecture. At this point, this paper was published 2012. It was, to me, a fundamental study showing that you can have alterations in mood and learning, drastic changes, without massive changes in circadian functions or sleep. Previous data in some way established a linear connection between light, circadian disruption, sleep alterations, and finally, mood and learning deficits. So in this case, we have a very interesting scenario where sleep and circadian rhythms are preserved. Whether there are alterations or minor changes, we still don't know. And this is a really cool question that hopefully someone will explore very soon. It's also important to mention that this is under the T7 condition, right? So the same question applied to any light stimulation. So a deep understanding of sleep under these conditions, I think that it could be essential, particularly when we think in the long-term effects. All these alterations are acute changes that were measured immediately after the T7 exposure. We have no idea what's happening in the long term and whether sleep could be one of the consequences of this irregular light exposure.
Okay, thank you very much for your answer. And now I stopped being unpolite. And Renske, can you please take over and probably take some questions from the chat, please?
Yes, of course. Thank you, Diego. Obviously, it was a great presentation. So a couple of questions came up.
Do you want me to stop sharing the screen or?
No, it's okay. It's okay. Whatever you prefer. Yeah. So one of the questions is actually when the light is given and it's irregular, is it LED light or is it anything else?
Under the, wait.
Yeah, I think under the irregular, under the T7 cycles or the T6 cycles.
Let me see if I could find an actogram. No, it's not here. Okay, so this T7 cycle, it presents brief exposure to light and dark, 3.5 hours. As you can see in this model, this is a 24-hour cycle. Animals are under this LD cycle. So the first few days, there is an abnormal light exposure, of course, because they received light and dark at different phases of activity, and it's hard to predict the onset of activity. However, what we found is that after 2 or 3 days under this condition, there is a robust circadian activity. In some way, the activity of animals is completely dissociated from the light-dark cycle. And the onset of activity can be easily predicted based on previous days, independently of the light exposure. As you can see in this model, we are constantly changing the light-dark phases. And the animals show these robust circadian responses. And in some cases, some days, the onset of activity correlates with light periods. In some cases, it's pure darkness. What we did, I didn't mention it here, but it's important to mention, is all the behavioral analysis or any measurement that we are doing here is after exposing animals to constant darkness for at least 1 day to avoid any direct effect of light on these responses.
Yeah. Okay, and I think more specifically, the light that you do use, is it LED or is it something different?
In this experiment, we use fluorescent lights.
Okay, got it. Perfect.
Yes. As I mentioned, it could be fantastic to explore different lighting conditions, intensity, spectrum. Yes, we don't have answers yet.
Yeah, it's only so much you can do, right? Yeah. So the next question, how specific is the effect of the T7 cycle for e.g. if on T6 or T8, do you see partial disruption? And what about different cycles like T14 or T21?
Okay, that's an interesting question. Honestly, I don't know. It's an interesting possibility. it could be nice. I think that the key point here is whether these cycles are forcing animals to follow the light-dark cycle or not. Mm-hmm. So the beauty of the T7 cycle is that animals are completely dissociated from the cycle. So you can see the onset of activity that it's not following light or dark pulses. So maybe, I haven't tried, and I don't know if people tried this before, but maybe different cycles like T9 or 5 could force animals to follow light-dark pulses. So at the end, you don't have circadian rhythmicity. You have an animal that is just following the environment. In this case, it's so difficult to understand what is the direct effect of light versus the circadian disruption. What Tara did here is to prove that you can dissociate these two functions, the direct effect of light versus the circadian component. And I would say that exploring different cycles is also really cool, always considering conditions where animals have robust circadian activity that is not following the light-dark cycle.
Right. Okay. Sounds good. Next question. You show that the PHB is involved in deleterious effects of irregular light-dark cycles on mood. Do you expect that the PHB is also involved in the positive effects of light treatment in, for example, depression?
These are fantastic questions. Unfortunately, I don't have answer for any of them. It's really interesting to think whether acute changes in brain circuits can be directly associated with behavioral outputs. Unfortunately, we don't have right now really good tools to evaluate deficits in mood. As I showed you here, we use the forcing test, which is an interesting test, but what we could see here is whether the animals are showing drastic changes. It's very hard to get like a dose response curve with this kind of test. Right. So we are seeing whether the animals show massive and significant changes or if there is no effect. Thinking in the beneficial effect of light, I'm thinking, and going back to the REN study, that we can consider light as part of the enriched environment condition. So maybe what we are doing with dim light is causing poor environmental conditions that then we can restore with better lighting conditions. So that's a different question. Regarding the PHB, we found that the chronic changes affect the function of the cells, the expression of PER2, but this was always after the exposure to these chronic periods of irregular light. We don't have information about what's happened at the beginning, and we don't have information about the acute effect of light.
Okay, thank you. Next question. Can the possible pathways for cognitive functions be different based on task nature and task complexity and temporal characteristics of light? And you touched upon this briefly already.
Okay, that's a great point. Let me go back to the model. So, as shown here, we know that light connects with the SCN and many areas in the thalamus. And from there, we can have multiple pathways by which these centers are connected with the hippocampus. In our study, we found that alterations in SCN wrote this irregular information to the hippocampus, affecting learning functions. How is this happening? We have no idea. Again, there are multiple pathways. If you checked, if you starting from the SCN and assuming 2 or 3 synaptic connections, you could have many brain circuits by which the SCN can affect the hippocampus. In the REN study, they characterize a circuit that it's involved in mediating the positive effect of light. But again, they have one target, that is the geniculate complex, and then they proposed in an elegant model that through the Raynaud's nucleus of the thalamus, the information finally reaches the hippocampus. But we have the same problem. There are multiple connections in between. And in these connections, we can speculate that any aspect of the environment could also impact these responses. Mm-hmm. For example, when we talked about mood, what they did was expose animals to a chronic model of stress, mm-hmm. which is obviously affecting arousal and brain state in general. So if we think about the cognitive effect of these models, it's completely possible that these alterations are also affecting these circuits mm-hmm. that are also involved in the effect of light on learning functions. Mm-hmm. So my point right now is we should focus on the first steps, mm-hmm. characterize what are the different pathways by which the retina can drive information to brain areas that finally affect the hippocampus. And possibly these areas are all interconnected. Mm-hmm. So, and going back to the previous question, the integration of this information, time of day, spectrum, and different variables could define this balance between positive or negative impact of light.
Right, right. So a lot to be done, I guess.
Definitely.
Yeah. Okay.
Yeah,
so
you can see all these studies were published in the last 3-4 years. So this is a really new area of research.
Yeah, it's a very developing field, and that's, I guess, so exciting because, you know, a lot to be done and a lot will be done as well.
Yeah, I would like to mention also that I said before that previous data show that alterations in circadian functions can then lead to mood and learning deficits. Mm-hmm. All these pathways are not saying that this is not correct. Mm-hmm. This is showing different level of complexity. Mm-hmm. Of course, changing lighting conditions can drastically affect sleep, can drastically affect circadian function, having massive effects. Yeah. In addition, light has direct effects. Mm-hmm. So it's really important to think about these light treatment protocols or whether light is present at the wrong time of the day without causing massive circadian alterations, but still having direct effect on behavior.
Yeah, very good. Yeah. And I think this question kind of ties into it because it's all speculation. So do you think the IPRGC projections to the VLPO are a potential circuit for changes in sleep, for example?
Absolutely. The VLPO, it's a really cool area, and how this circadian system is also affecting VLPO, it's something that we are just started to understand. Mm-hmm. And I would like to go back to the introduction where I showed that a single IPRGC projecting to the SCN also has collateral projections. Mm-hmm. So another interesting possibility to think is whether this massive and diverse innervation to multiple areas can have an overall effect on the connectivity between these different regions, affecting not only circadian function, but also circadian component of other functions such as sleep or cognitive functions, for example.
Right. Yeah. Very cool. Yeah. Next question. Do you think that the IGL pathway may also be an explanation in why timing of food intake may influence circadian entrainment when light-dark cycle changes are not very prominent in organisms, people living in a regular 24-hour cycle?
Yes. Okay, that's a really interesting question. Our studies used environmental conditions where food was the only time cue. As I mentioned before, there is no data in the literature showing that there is in fact integration of time cues. We know this from the behavioral output. When we manipulate feeding conditions with LiDAR cycles, it drastically affects mouse physiology and the behavior. Where is this integration happening is still not known. This is, in my opinion, the first demonstration that there are areas that can actively integrate. And here we use extreme conditions where we removed IPRGC innervation. There are still open questions on whether this IPRGC projection can actively modulate these circuits. Mm-hmm. In this case, it would be important to understand how the integration is also comparing the power of different time cues, whether you are exposed to a regular light-dark cycle or abnormal feeding condition, which one is taking control. Mm-hmm. So that's the beauty of the idea of an area that can integrate, because it's not only integration, it's sensing what is changing in the environment, and based on these changes, anticipate and coordinate the circadian function as a whole, as a system.
Right. Yeah. And final question, I think. So you had a slide on metabolic alterations in shift work. And currently the WHO has classified night shift as a carcinogenic hazard. So the question is, if circadian rhythm disruption is the fundamental problem, would that not be the health hazard?
Can you repeat the last part, please?
So I think the question is, if circadian rhythm disruption in itself is the fundamental problem, is that the health hazard, as opposed to night shifts being classified as the health hazard?
Okay. Let me see if I understand. My opinion, the way I see this, the problem is when we talked about shift work, we have so many variables, so many things that are changing. In my research, I focused on the effect of light. Mm-hmm. And that's why we use this strong and robust model where we removed IPRGCs. Mm-hmm. Obviously, real life is much more complicated, and you have different conditions where people that work at night is clearly receiving abnormal light exposure. But they also have alterations in many, many variables, feeding conditions, social interaction, and the stress that is associated with this. So I think that under these conditions, it's not just one pathway, it's possibly the combination of multiple pathways. As we were talking before, abnormal light could have effect through the circadian functions, but also direct effect, affecting in mice, for example, the PHV or SCN and learning functions. In addition, the direct effect of light on circadian functions and sleep are causing several problems. So at the end, what we have is a list of symptoms that define the alteration. And I think, as I mentioned before, that understanding the pathways that are affected, particularly if they are parallel pathways, could give us a better understanding of the symptoms, the pathology associated with the person and not the pathology in general. And based on this, work on better treatments to tackle the specific problems and not the general description of the pathology.
Yeah, so form of personalized medicine, let's say.
Exactly.
Perfect. Yeah, I think with that we answered all the questions, or you answered all the questions in the chat window. So thank you very much. Yeah, thank you very much for your talk in general. I think it was great. This was also the last talk of the SLTBR webinar series for now. Yeah, I hope to see everybody at the virtual conference. And thank you again, Diego. It was great.
Thank you very much. Here is my email in case you have any question, you can contact me. Thank you.
Perfect. Thank you.
Okay.